Hi students, welcome to SPSVERSITY. In the previous session, we discussed about the DNA structure, the search of DNA material, and the stability of DNA, and the experiments related to DNA. So now we are going to discuss about DNA replication. So when I come to DNA replication, so in this DNA replication, DNA, another two DNA are formed. When does it take place? When this process occurs. So this process occurs during cell division, so which is an important process for growth of an organism. In cell division, specifically during S phase of cell division. S means synthetic phase of cell division. So during synthetic phase of cell division, DNA replication takes place. Okay. So from one DNA, two DNA are formed. That means on each replication, the amount of DNA becomes doubled. So whatever the DNA are having daughter cells, that is coming from the parent DNA. That is, the parent DNA will form, that is, will be given to the daughter DNA on replication. So that's why the daughter cells have the same genetic information. So that is the reason why the daughter cells will have the same genetic information. So let's see how this process DNA replication takes place. So this DNA replication, actually it is proposed by, that is, the DNA replication is proposed by the person, that is Watson and Crick. So he has told that the life sentence, that is, if you take this, it has not escaped our notice that specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Copying mechanism means here it is nothing but DNA replication. How it is proved? On what basis, that is, we can prove the DNA replication takes by the specific method? To prove this mechanism of DNA replication, two people called Mendelssohn and Stahl designed an experiment. What is this experiment? We'll see. In 1958, so Mendelssohn and Stahl used a bacteria called E. coli as an experimental tool. And this bacteria, which is taken as an experimental tool, initially it was grown in N15 medium, that is N15, a radioactive substance which is grown in the N15 medium. After that, the DNA is extracted from this E. coli and on centrifugation by using cesium chloride. That is, by using cesium chloride and the principle, that is density gradient, the DNA is extracted from these cells. The DNA separates from these cells as the density of DNA is equal to the density of cesium chloride solution. Okay. So now they have extracted a DNA with N15 labeling. That means the DNA which is extracted from E. coli on centrifugation, it is having labeling, that is N15 DNA. So this N15 DNA, now they have taken for the process of next step of experiments. In the next step of experiments, this N15 DNA was grown in N14 medium. So here the point should be noted is E. coli cells with only N15 in their DNA were transferred to N15. That means the DNA which is present in E. coli has a red labeling, that is N15. So what are the requirements for this? What are the requirements for this experiment? That is bacteria E. coli, nutrient medium with isotope, that is heavy nitrogen, that is N15, nutrient medium with normal nitrogen, that is N14, high molar solution cesium chloride and ultracentrifuge, that is which works on the principle of density gradient diffusion. So these are the requirements of this experiment. What is the result of this experiment? The result of this experiment says that half of the parent strands of DNA are conserved. That means if you take a DNA, one DNA, suppose if you take one DNA, and in this DNA there are two strands. So one strand belongs to parent, that is parent is, it comes from parent. Other strand is the new strand. That means half of the strand is, half of the DNA is parental DNA, half of the strand is newly synthesized. So this method of replication is called semi-conservative method of replication. We will see the experiment here. So they have taken E. coli with N15 labeling. So the DNA of E. coli multiplies for this, it undergoes multiplication, that is replication for every 20 minutes in N14 medium. It is kept in N14 nutrient medium. When it is kept in N14 medium, after 20 minutes, 2 DNA has come. So the 2 DNA, if you see carefully, this is the parent DNA which is N15. This is newly synthesized DNA which is N14. That means 50% of the DNA, that is one strand of DNA is old, one strand of the DNA is new. Parent one is old and newly synthesized. The same thing to this DNA also. This is N15, this is N14. So this type of replication is called semi-conservative method of replication. Again, these 2 DNA after 40 minutes, they undergo replication. So total we get 4 DNA. So in this 4 DNA, 2 are of having N15 and N14 and 2 are of completely N14. So what this proves? That is, it proves that the method of replication is semi-conservative method of replication. So what is meant by semi-conservative method of replication? Semi-conservative means the newly synthesized DNA strand, has half of the DNA is old and half of the DNA is newly. Old means the parent strand and the new one is the newly synthesized. So here we will come to the DNA replication process. So what is the machinery required and the enzymes required for this DNA replication process? So it requires enzymes, as well as there are many enzymes required for this, like enzyme called helicase, which will separate the 2 strands of DNA. Primase, which synthesizes short RNA sequences called primers, where it is starting point of DNA synthesis. DNA dependent DNA polymerase, where they will synthesize, that is, they will catalyze the polymerization of new DNA in the direction of 5 to 3. DNA ligase, it joins the DNA fragment and the parent DNA. Parent DNA means the DNA which undergoes replication. So we will go into the steps of DNA replication. So DNA replication starts at a specific point on DNA, that is in a specific region of DNA, which is called origin of replication. In short form, it is called ORI, origin of replication. So the point where the DNA replication starts, it is origin of replication. So from this point, the replication will start. So first, the step is unwinding of DNA takes place. So due to unwinding, both the DNA strands get separated. Due to unwinding, both the DNA strands get separated. So both the DNA strands are separated means here the DNA will be, that both the strands of DNA will be coiled with each other. Due to this helicase enzyme, both the strands of DNA get separated. So this is one strand, this is one strand. So each strand acts like a template. So each strand acts like a template. After separation of DNA, a short stretch of primer is synthesized with the help of enzyme primase at 3N. Suppose this is 3N, understand it is 5N, this is 5N, this is 3N. Okay. So a short stretch of primer is synthesized at 3 end of template. Okay. So here the primer acts as a stone, stepping stone for replication starts. That is, it's a starting process of DNA replication. Then later on, next enzyme plays a role, that is DNA dependent DNA polymerase. So where it binds to the template strand and starts polymerization. So here polymerization means it go on adding the complementary nucleotides in 5 to 3 direction. The new strand is always synthesized in 5 to 3 direction. Okay. So here you can see the diagram of the replication point, that is origin of replication. So the point where the replication takes place, you can see the two DNA strands are separated apart, which looks like a inverted Y shape. So this Y-shaped structure is called replication form. So this Y-shaped structure is called replication form. So here you can see, so this is a replication form. So here the parent strands, these are parent strands which has the anti-parallel in direction 3N to 5N, other is 3N to 5N. In one strand we can see there is a continuous synthesis of new strand in 5 to 3 direction. So continuous synthesis is going on. That is, with the help of DNA polymerase, DNA dependent DNA polymerase, you can see the polymerization takes place. So it is called leading strand. It is called leading strand. Okay. And when you come to other strand, in other strand there is a discontinuous synthesis. That is, it is synthesized in the form of fragments. So these fragments are called Okazaki fragments. And these Okazaki fragments later they are combined by the enzyme DNA ligase. They are combined by DNA ligase. And this strand where discontinuous synthesis takes place, it is called lagging strand. It is called lagging strand. So in one strand there is a continuous synthesis, and in another strand there is a discontinuous synthesis. In discontinuous synthesis, it is synthesized in the form of short fragments. They are called Okazaki fragments, which are joined by DNA ligase. And this process continues till the entire DNA is copied. So that means till the entire DNA forms 2 copies of DNA, this process will continue. So here you can see the central dogma which is proposed by the crick. This DNA on replication will form DNA, on transcription it will form RNA, from translation RNA will form protein. By reverse transcription, from RNA DNA is formed, which is seen in retroviruses, where in retroviruses RNA is a genetic material, where RNA will form, that is DNA, by using enzyme reverse transcriptase, which is seen in retroviruses like HIV. The enzyme name is reverse transcriptase, and this process is called reverse transcription. Okay. So after replication, that is in replication, both the strands will act as a template. A next step which plays an important role in the molecular biology is the DNA transcription. Okay. What happens in transcription? So whatever the information stored in DNA, it is copied to RNA by the process called transcription. And the end product of the transcription is RNA. RNA, which is formed in further process by translation, it forms proteins. So here, whatever the information stored in DNA, ultimately it leads to formation of proteins by 2 steps. One is by transcription, later on by translation. And here what I discussed already, in few viruses DNA is formed by reverse transcriptase, where like in HIV the genetic material is, that is RNA. From RNA the genetic material is formed, that is RNA forms DNA, which is called reverse transcription. So now we'll see what is meant by transcription. What happens in transcription? What are the process takes place in the transcription? So in transcription, whatever the information, genetic information present in DNA is passed on to, that is copied to RNA, which is called transcription. So in this process, the complementary relationship plays an important role. As we already know, in DNA, A is always complementary to T with 2 hydrogen bonds. G is always complementary to C with 3 hydrogen bonds. But when it comes to RNA, in RNA thymine is absent. In the place of thymine, uracil is added, which U represent by capital U. And next thing is, in RNA, that is in most of the RNA, it is single stranded. It is only one stranded. So that means in transcription, only one strand of DNA, it is only one strand of DNA acts like a template, not both the strands. One strand of DNA acts like a template. Others DNA doesn't act like a template. What is the reason for this? Suppose if both the strands of DNA work as a template, we'll get 2 different RNAs, which will form 2 different proteins, where the process becomes, the next steps becomes very complex. That is the reason why only one strand of DNA acts as a template. So first we'll discuss about where transcription takes place. At what point in DNA transcription takes place? In DNA, the segment of DNA, that is the part of DNA where the transcription takes place, it is called transcription unit. In a transcription unit, it is nothing but transcription unit is nothing but a sequence of nucleotides. In a transcription unit, there are three parts: a promoter, structural gene, a terminator. What are the enzymes required for this? That is, the enzyme required for transcription is DNA dependent RNA polymerase. And DNA dependent RNA polymerase, there are two factors, that is sigma factor and rho factor. Sigma factor to start the process, rho factor to stop the process. So first, when you come to the diagram of a transcription unit, a transcription unit has three parts, that is a promoter, a structural gene, terminator. So in this transcription unit, only one strand acts like a template, that is, this strand acts like a template strand, and other strand is called coding strand. So in two strands of DNA, only one strand of DNA will act as a template. Other strand of DNA is called coding strand. And the parts of a structural transcription unit is promoter, structural gene, after that terminator. So here when we come to the terminology, template strand, that means the strand of DNA which will be transcribed to, that is, which is copied to in the form of mRNA. Coding strand, which is not copied, that is, which is not used in transcription process. Promoter, the point of, that is, replication, the region of replication, transcription, where the enzyme Rna Polymerase binds. That means the process of transcription starts from promoter. Next, structural gene. The structural gene means from which whatever the information present in structural gene, it is copied to RNA, which is present between promoter and terminator. And the end part is terminator, where the point of transcription, that is, this is the point where transcription will end. It is called terminator. So here when we come to this process of transcription, we know that the enzyme plays an important role, that is Rna polymerase, which plays an important role. It is DNA dependent Rna polymerase. So when we come to the Rna polymerases, when we come to specifically the eukaryotes, there are three types of Rna polymerases: Rna polymerase 1, Rna polymerase 2, Rna polymerase 3. Rna polymerase 1, which is found in nucleolus, will help in formation of rRNA. rRNA means ribosomal RNA. Rna polymerase 2 will form mRNA, messenger RNA. Rna polymerase 3 will form tRNA, that is transfer RNA. Here if you take ribosomal RNA, that is, it is insoluble RNA. Okay. So here when you take this, the point, that is, when you take the point, that is in DNA, the point where transcription takes place, that is in promoter, specifically in prokaryotes, there is a sequence called TATATG sequence, which is called Preno box. That is called. Next is Tata box, which is found in promoter regions of eukaryotes. That is, in promoter region of prokaryotes, this is TAT, whereas in prokaryotes it is TATATAG. It is called Preno box, where it is called Tata box. Okay. So here when you come to the point where when you come to the gene, what is actually a gene? So why we are going to discuss now at this point. So a gene is the basic unit of inheritance where it will express the character. So how it will express the character? So previously Gregor John Mendel, by his hybridization experiments, he called them as factors. Later on, Johansson, he called them as a gene. The meaning of gene is to give birth to. And later on, Biddle and Totem discovered the genes and the control of production of enzymes. That is, he proposed, Biddle and Totem proposed one gene, one enzyme hypothesis. That means from every gene, one enzyme is synthesized. Later on, Avery, when you come to Oswald T. Avery, announced that DNA is the substance for responsible heredity. In 1950, Watson and Crick, so Watson and Crick discovered the structure of DNA, that is double helix structure of DNA. Later on, that is when you come to Benger, proposed the modern concept of gene working on equivalently to the. According to Benger, what is a gene? A gene is a cistron, recon, muton, replicon. What are these? Cistron, recon, muton, replicon. Cistron means it is a structural gene where it can undergo transcription. That means it codes for a specific protein, means it can undergo transcription. It is coding for a specific protein, means the gene where which can undergo transcription, it is called cistron. Recon, it is the unit of DNA that undergoes recombination. That means which can form new recombinant features. Muton, this is the unit of DNA that can undergo mutation, where it can undergo changes. Replicon, it is the unit of DNA that can undergo replication. So here, according to Benger, he has given a gene is a cistron, a recon, it may be a muton, it may be replicon. Cistron is the structural gene. Recon, it is a gene where it can undergo recombination. Muton, where it can undergo mutation. Replicon, where it can undergo replication. Okay. So here when we come to the part of RNA, that means by the end of transcription we are getting RNA. The end product is RNA. There are three types of RNA: mRNA, messenger RNA, tRNA, transfer RNA, rRNA, ribosomal RNA. So mRNA, tRNA, rRNA has their role during protein synthesis. mRNA provides, it acts like a template by which the amino acids sequence is decided. tRNA brings, that is, amino acids which are required for formation of proteins. Ribosomal RNA plays a catalytic role. Catalytic role means for formation of peptide bond between the amino acids during protein synthesis. So here we have discussed about, that is, three enzymes are present. Rna polymerase 1, 2, 3 are present. What about prokaryotes? In prokaryotes, there are only, that is, there is only one DNA dependent RNA polymerase. And this RNA polymerase will synthesize the new RNA in 5-3 direction. We'll see what are the steps of transcription. There are three steps, that is, initiation, elongation, termination. So initiation means where RNA polymerase binds to promoter region. So for this, to start the process of transcription in RNA polymerase, sigma factor plays an important role. In elongation, the RNA polymerase will copy the information present in structural gene. That is, the copying of structural gene takes place in the elongation process, where the complementary base pairing is formed. Here complementary base pairing means, suppose in DNA if there is adenine, it should form thymine uracil, but in RNA uracil is present. In DNA if there is guanine, in RNA it will form cytosine. If there is thymine, it will form, that is, adenine. Here if it is C, cytosine, it will form guanine. In complementary base pairing, on the principle of complementary base pairing, a new RNA is formed from DNA template strand. So once the transcription is done, that is, transcription of structural gene is done, at the end of transcription, Rho factor binds to the enzyme, and this Rho factor will leads to, that is, the RNA polymerase is released from DNA template. When Rho factor binds to enzyme, RNA polymerase leaves the DNA template. So that what happens? The process of transcription stops. So here you can see the process. So the process starts from promoter. So in RNA polymerase, the sigma factor helps in initiation of the process. So here initiation takes place. The structural gene is copied. That is, it copied means the DNA in the structural gene will form RNA, which is called elongation. Later on, when it reaches to terminator, the Rho factor binds to RNA polymerase, where RNA polymerase gets detached from the DNA, which is called termination. So here, if you see this process, the new strand, that is RNA, which is synthesized, it is synthesized in new RNA is synthesized in 5n to 3n direction. So what are the difference between the process of transcription in eukaryotes and prokaryotes? In eukaryotes, there are three types of RNA polymerase 1, 2, 3. RNA polymerase will form ribosomal RNA, that is 28S, 18S, 5.8S. RNA polymerase 2 will form RNA, hnRNA. RNA polymerase 3 will form tRNA, 5S RNA and snRNA. And the second one is, whatever the primary transcripts are formed, that means as and when the process of transcription is over, they are called primary transcripts are formed. They cannot be functional. These are non-functional. So they should be undergone process. After that only they become functional. What is that process? In primary transcripts, that is, they contain parts like exons and introns alternately. So here introns should be removed and exons should be joined. It is called splicing. That is, along with three other enzymes, there is another complexity, that is, splicing takes place. What is this splicing? So in this splicing process, whatever the RNA is present, in the RNA which is formed, the primary transcript RNA, that is called hnRNA, heterogeneous nuclear RNA. So the primary transcript which is formed in eukaryotes, it is called hnRNA, which has exons and introns alternately. Here you can see this is intron, this is exon, which was present alternately. So the first step here is, the first intron should be removed. Then the exon should be joined. So, for removal of this introns, spliceosomes will play an important role in the removal of introns, and later on the exons are joined. After joining, that is after splicing of RNA, the next step is capping. In capping, at 5N guanine is added. That is called capping. This is capping, where the 5N, that is at 5N, that is the guanine is added. And at 3N, adenine polyatyl is added. It is called polyadenylation, so which is called tiling. So, in the processing of hnRNA, there are 3 steps: splicing, capping, tiling. So, in this process, by the end, what we get, that is mRNA. That is hnRNA on processing. Of processing means the processing of these 3 steps: splicing, capping, tiling, will get ultimately mRNA, which is a functional RNA. hnRNA is not a functional RNA, it is non-functional, whereas mRNA is a functional one. So, splicing, capping, tiling are the steps in the processing of that. In capping, at 5N, methylguanosine triphosphate is added. That is 5-GPP is added. In tiling, adenylation, polyadenylation takes place, where adenylate is added, 200 to 300, which is a 3N. So, by the end, we got mRNA, that is messenger RNA. So, what you are seeing, this is a messenger RNA. It is 5N, it is 3N. So, after 5N, that is after cap, that is after before tile, there are untranslated regions, ETR. These are called ETR, untranslated regions. In between these two, there is a protein coding sequence. It is a protein coding sequence. So, here we will see what mRNA is. mRNA is nothing but a messenger RNA. As already discussed, messenger RNA. In messenger RNA, what message it carries? It carries message that to carry out protein synthesis. In what form the message will be present? Anyway, in RNA there are 4 types of nitrogen bases. It is adenine, guanine, cytosine, uracil. No thymine. So, these 4 types of nitrogen bases with nucleotides, which should represent 20 different amino acids. In what form they will represent? So, this is called the genetic code. The relationship between the RNA and the DNA, it is called genetic code. What is this genetic code? What code? So, in RNA, the information is present in the form of codons. What is this codon? Each codon will represent each codon will represent 1 amino acid. So, here a person called George Gamow has proposed that there are only 4 bases that has to code for 20 amino acids. That means 4 nitrogen bases should represent 20 different amino acids. So, what combination they should have? In order to code for 20 amino acids, the code should be made up of minimum 3 nucleotides. Okay. It should be of minimum 3 nucleotides. Okay. That means 3 nucleotides means 4 to the power of 3. That means if you multiply 4 multiplied with 4, you get ultimately 64 codons. Ultimately, what George Gamow gave is, that is, A codon on mRNA is a triplet codon. Triplet codon means it has, that is, triplet codon means it has, that is, 3 nitrogen, 3 nucleotides. Each and every codon has minimum 3 nucleotides. So that's why it is called triplet codon. So when you take the total, that is 4 nitrogen bases, that is total, if you take triplet codon, that is total, we will get 64 codons. Why can't it be a doublet codon? Suppose if it is a doublet codon, so what we'll get, a doublet codon means 4 multiplied with 4. How many will get? 16 codons. So total there are 20 amino acids. Suppose if it is singlet one, only one, then only will get 4 codons. How can they represent 20 amino acids? So the confirmation that is a codon is a triplet codon. And the first illustration of codon was done by Hargobind Khurana. It is done by Hargobind Khurana, Marshall Network and others. Later on, the cell-free system of protein synthesis helped, that is, to find out the codons. The cell-free system of protein synthesis was done. So, by Nirenberg, Khurana, Hawley and Nirenberg received Nobel Prize in medicine for their work. So, the output of this, what it says is the codons present on mRNA or triplet codons which has, that is, 3 nucleotides. So, this is the table of codons where we are seeing different codons here. Here total you can see, that is 64 codons. In codons there are 3 nucleotides, that is the first letter, the second position and the third position. So total we are having 64 codons, and each codon represents one amino acid. So these are, which are written on the right side, these are names of amino acid: valine, methionine, isoleucine, leucine, phenylalanine, serine, proline, threonine, alanine, tyrosine. Stop means these are stop codons where the process of translation will stop. Histidine, glycine, aspargine, lysine, aspartic acid, glutamine, cytosine, arginine, glycine. So like that, they will represent, that is 64 codons will represent different, that is 20 amino acids. What are the features of genetic code? In genetic code, as we already discussed, there are, as it is a triplet codon, there are 64 codons. In this 64, only 61 will code for amino acids. These are called sense codons. And remaining, what they will do, they will not represent any codon. That is, they are called stop codons or terminating codons. Once they come, the process of translation will stop. And one codon will code for only one amino acid. So here it is called, this nature is called unambiguous and specific. But whereas one amino acid, some amino acids are represented by more than one codon. That is, it is called degenerative. It is called degenerative. For example, if you take UUC, it will represent phenylalanine, it will represent phenylalanine. It is called degenerative. And the codons are read in contagious fashion. Contagious means we should write continuously. You should not use any exclamation marks, commas, full stops while you are writing. The codon is universal with a few exceptions. And here AUG is a codon which is starting codon, which has dual functions. That is, it is called Initiator Codon. Initiator means where the process of protein synthesis starts, AUG, which codes for methionine and is also acts as a initiator codon. So these are the salient features of genetic codon. Then what is meant by anticodon? So anticodon means that is the complementary of codon that is called anticodon, which is present on tRNA. Codon is present on mRNA, whereas anticodon is present on tRNA. For example, if it is AUG, the anticodon will be complementary UAC. If it is UUU, it will be AAE. If it is CGA, it will be GCU. So here you can see this is mRNA where codons are present. So this is the anticodon that is which is present on tRNA. This is the tRNA which brings amino acids during protein synthesis. Okay. So here in the process of the molecular biology, we know very well mutations will take place. So mutations means a sudden change in the gene or chromosome. A sudden change in the gene, that is a change in the gene due to single base mutation, it is called gene mutation. If the change occurs in chromosome, it is called chromosomal mutation. So here in chromosomal mutation, where we discussed about the Klinefelter syndrome, Down syndrome, and next we discussed about that is Turner syndrome. But when we come to gene mutation, in this there are frame shift mutations and substitution. What is this frame shift and substitution? So here in gene mutation, just change in one or few base pairs will lead to mutation. We'll take an example, that is, example is sickle cell anemia. So here remaining chromosome number remains shape and change. But here you can see there is a change in gene where we cannot be identifying any karyotype. Karyotype means the diagrammatic representation of chromosomes. We cannot identify, but it will follow Mendelian pattern of inheritance. What is this frame shift mutation? So in this frame shift mutation, what happens here? That is, it may be due to insertion or deletion of bases. It may be due to insertion or deletion of bases. So just for example, if you see, that is the cat saw the rat. Just for example, take it as a mRNA. Assume that is each is one base. Suppose in the before just cat, if you add A, if you add A, what will happen? That is, sequence of codons will change. That is, the nucleotides will change. See how it is changing. Like this, if we add or delete any nucleotide base, it will change. Here you can see one base pair may be changed to another, okay, which results in sickle cell anemia. It's not only insertion or deletion, sometimes you can see substitution. So substitution means in the place of one nucleotide, that is, it is replaced by another one. For example, substitution of thymine with adenine. Obviously, it will change the sequence of the coding numbers. For example, if we take this one, here you see the DNA sequence is GAG CTC. It will form RNA GAG, which will form glutamic acid. But in the place of A, if it is replaced by thymine, what will happen? It will form GUG, which will codes for value. That means one single base pair in a DNA, if it changes, it will change the, even it may change the amino acid also. In the place of one amino acid, another amino acid is formed, which results in the phenotypic change. Which results in the phenotypic change. So now, we'll discuss about the next step after transcription, that is translation. What is meant by translation? Translation is the polymerization of amino acids to form a polypeptide. Poly means many. Many peptide bonds are formed between amino acids. So, it is the process by which protein synthesis from mRNA. So, mRNA on translation, it will form a protein. So, what are the requirements for translation? So, mRNA, tRNA, ribosomes. If it is in prokaryotes, 70S. If it is in eukaryotes, 80S ribosomes. Amino acids which are present in amino acid pool. And we require amino acid tRNA synthetase and enzyme. Proteins, that is initiation factor, elongation factor, releasing factor, and magnesium is Mg2+. So, in this process we require, that is mRNA, tRNA as well as rRNA also, that is which acts like an enzyme. We will come to the structure of tRNA. The tRNA is a molecule which looks like a hairpin structure. Okay. It is a 3 clover leaf model. So, if you take this model, the tRNA which looks like a clover leaf model, one end of the tRNA it is called anticodon end, and other end, that is opposite to anticodon end, it is called amino acid binding site. It is called amino acid attachment site. It is called amino acid attachment site. Here you can see clearly the structure of tRNA. So, this is the point where amino acid attaches. It is called amino acid attachment end. It is called the anticodon end, where the anticodons will bind. So, at one end it attaches to mRNA, at another end it carries, that is amino acid. It is called adapter RNA. Okay. So, this tRNA plays an important role in carrying out the process of protein synthesis. That is, they will carry amino acid during protein synthesis. So, what happens during this protein synthesis? So, during this protein synthesis, what are the changes taking place? What are the steps? Okay. The first step here is, whatever the amino acid is represented by the codon, this amino acid binds to the RNA, which is called charge chain. So, when amino acid attaches to tRNA, it undergoes charge, that is, it is called charge. There is a two-step process. In this, the enzyme which plays an important role, that is amino acid tRNA synthetase. First, initially, it requires energy, that is, it binds to ATP. Okay. It will form, it binds to ATP to amino acid to form amino acid AMP complex. Are linked by high energy form, where the two phosphates of the phosphate is released. So, here amino acid binding with ATP, it will form, that is AAMP, adenosine monophosphate. Then the amino acid binds with tRNA. Now, tRNA gets a charge. Now, it is called charged tRNA. Now, it is charged tRNA, that is amino acid with tRNA, amino acyl tRNA. It is called amino acyl tRNA. And this entire process, the charging of tRNA, it is called amino acylation of tRNA. It is called amino acylation of tRNA. After amino acylation of tRNA, the next step, that is here, ribosomes play an important role. During protein synthesis, whenever mRNA is formed, to mRNA, smaller subunit of ribosome is attached first and later on larger subunit is attached. In larger subunit of ribosome, there are three. There is acylation site, peptidyl site and exit site. Okay. So these are the sites where the tRNA will attach. These are the sites where tRNA attach. That is, if you take a ribosome structure, in ribosome there is smaller subunit and there is a larger subunit. This is, and if you take this one, this is an 70S ribosome. 30S smaller subunit, 50S larger subunit. So here, what happens? So here, mRNA acts as a translation unit. So the sequence of RNA that is having the codons, starting codons and stop codons, it is called mRNA. You see the structure of mRNA. It starts with untranslated regions. So untranslated means on either side of the mRNA there are untranslated regions which doesn't undergo any translation process. In between this there is a translation unit. So which starts with initiator codon, that is it starts with initiator codon AUG. And it starts with, that is stop codons UAG, UAA, UGA. What happens during initiation? In initiation, that is the initiation factors binds to smaller subunit of ribosome. Then later on, mRNA attaches to the smaller subunit of ribosome. And later on, that is after this, the initiator codon, which lies on P site, and the second codon lies on A site. So the first initiator amino acid tRNA gets attached to P site. That is, later on larger subunit attaches to here. That means whatever the initiator amino acid tRNA, that is the first tRNA which brings the methionine, that is AUG, which codes for AUG. It attaches to P site. It is E site, it is A site. So this is AA1 amino acid 1. This is AA2 comes and joins here. That is the translation, that is next tRNA brings to here. That is A site. Next step is elongation. In elongation, the second tRNA attaches to, that is it comes and attaches to A site. That is, in P site and A site there are amino acid. Between these two, peptide bond is formed. So as and when the peptide bond is formed between two amino acids, the bond between first tRNA and methionine breaks. That is, tRNA is released. Here you can see the tRNA is released. So ribosome moves to next codon. That is, ribosome will move in this direction, 5, 2, 3, N. So first tRNA will exit from E site, exit site. The process will continue until it reaches stop codons. Stop codons means the final step is termination, where the tRNA does not have, that is the tRNA doesn't break any amino acids. Stop codons means on mRNA, that is whatever there are there, that is UAA, UAG, UGA, which represents, that is stop codons. Okay. During this process of termination, protein releasing factors, that is releasing factors binds to ribosome and cause release of all components. That is, the ribosomal units get separated, mRNA and polypeptide. This process will stop. So by the end, the process of translation stops when they come in for us these stop codons UAA, UAG, UGA. Thank you all. Let's follow us on social media, SPSVERSITY. We'll meet in the next session.